Well, I was going to, and are people hearing me? Yeah, I guess now. I was going to say what an honor it is, how proud I am to actually speak at this function. It's really, Alberta is doing incredibly well with innovation, and I'm lucky enough to serve on the AI Biosolutions Board of Directors, and I've just been impressed and impressed and impressed as time goes on. So I was going to start with that, but I think instead I'll start with saying how hard it is to follow Jackie, because I really feel kind of inadequate in several ways. But I'll try. In addition to starting companies and being a scientist, I'm also a physician, a neurologist, and my lifetime clinical obsession has been amyotrophic lateral sclerosis. Does anyone in the audience know what that is? Can you raise your hands? Okay, so about half or so. It's so everyone knows, of course, what Alzheimer's is. Everyone knows, of course, what Parkinson's is. But fewer people know what ALS is. It's another neurodegenerative disease, and I'll show you some information about that and how we might be able to approach it. So another thing I like to present with my talks, you know, that we're always required as academic researchers to put up our disclosures, our industrial disclosures. Well, that sounds so furtive, like you're trying to hide it and somebody's forcing you to bring it out. So I changed the word to engagement. It also gives me the chance to say that I actually founded 2 biotech companies in Canada, both of which are publicly listed. I have provided, I've licensed a technology to Biogen, also to CanGene, which has now been bought up by Emergent Biosolutions in the United States. And I serve on the scientific advisory board of Prothena Biosciences in the Bay Area. So I want to really summarize this in 2 slides. This is my intellectual passion, but if you understand these 2 slides, then you understand what I'm doing and why I'm doing it. I think we all learned in high school that proteins are a chain of amino acids, but maybe we learn in university or not that this chain of amino acids actually is folded into a precise 3-dimensional shape in order to achieve the function that it was designed for. So this doesn't have any function as a protein. It's just a floppy string. This has a function when it's been folded properly. It acquires its function. Maybe it stops there in our training, but over the past 10 years or so, it's becoming increasingly clear that proteins can become misfolded in a kind of propagated pathway, almost like, as Val Sim was showing, a chain of dominoes. One bad protein convinces the protein next to it to convert into more of the bad kind, et cetera, et cetera, et cetera, till all of the protein in a brain or a tissue is converted into this abnormal toxic form. Of course, that idea came from the prion world, from Stan Prusner's discovery, sanctioned by the Nobel Prize, that the prion protein is an infectious agent because it acts in this domino fashion, templated at the beginning or spontaneously misfolding or genetically determined. Once the seed of misfolded protein is formed, it gets propagated. But surprisingly, or maybe not so surprisingly, and Val mentioned this prion idea is starting to find application in common neurodegenerative diseases. There's a prion-like component to Alzheimer's disease. There's a prion-like component to Parkinson's disease. There's a prion-like component to ALS. There's a different protein in the starring role of all of those diseases, but the concept, the process is the same. We're also exploring the idea that protein misfolding may not be global or permanent, that there are regions of proteins that can partially unfold in a kind of dynamic process, unfolding, folding, unfolding, folding. And this may achieve significance in the treatment of cancer. And I won't go into a lot of detail about that, but I'll show you the second disruptive idea that my work and my companies are trying to advance. And that's the idea that antibodies are perfect golden bullets or silver bullets for these sorts of diseases. We're kind of all raised in the scientific world to believe that if we only turn it over to Pfizer, they'll find a small molecule that fits in a precisely defined pit or cleft or channel or something like that, a rigid target on the surface of a protein that can be bound by a small molecule, also itself essentially rigid. But for protein misfolding, that's the wrong pathway. Protein misfolding is characterized by large floppy regions of proteins, not a little defined pit or pocket, but a floppy region in which small molecules are essentially useless. But an antibody can be designed or selected to specifically bind to a misfolded portion of a protein. It's like, this is what antibodies eat for breakfast. Small molecules can't do anything about these floppy regions, but antibodies can. And there's an advantage also with antibodies that they may neutralize the toxicity of a misfolded protein. We know that they're essentially all toxic. They kill cells. They may block the propagation of misfolding. They may accelerate degradation of the misfolded protein. But there's a safety aspect to this idea as well, that if you're leaving the normal protein alone, sparing the normal protein, you don't interfere with the function of that protein. You don't have widespread whole body autoimmunity, all of the places where this protein is found. So we have, we now, based on these two ideas, have, okay. We have a vaccine under development for chronic wasting disease. As Val was mentioning, it's spreading like crazy in the wild. We don't have a treatment or prevention, but there is a possibility this vaccine against partially misfolded prion protein could provide that protection. We are exploiting at Amorphix, the company I started, Amorphix Life Sciences. The initials, of course, are ALS. We have a raft of specific immunotherapeutics for cancers which spare the normal cells. Again, this same idea of leaving the normal cells alone and only targeting the sick cells or the bad guys. We have a therapeutic for Alzheimer's disease in development by CanGene, which recognizes a specific immunologic target at the surface of A-beta oligomers. Do I have a few more hours? Okay, I guess so. And finally, we've recognized that superoxide dismutase 1, SOD1, there will be an exit test, is a potential target in the treatment of ALS. If we can stop the cell-to-cell spread of SOD1 propagated misfolding, then we might be able to arrest ALS. This technology was developed through Amorphix, but is now under development by Biogen. So quickly, in such a simple way, even I can understand it. Here's protein misfolding, a sick protein inside a cell, which is converting its neighbors, going to a neighboring cell, and converting those normal proteins and producing cell death. So if we have an antibody or a vaccine, the same process occurs, but it's blocked in the space between cells, and the nerve cell is protected. Just one more thought. Out of the Big Bang of the prion protein and prion diseases, there are now tie-ins for not only Alzheimer's, ALS, and Parkinson's, but type 2 diabetes, Huntington disease, amyloidosis, and even perhaps schizophrenia. It's a brand new world. And I just want to show you the old geek in the middle is me, but here's my young, spirited, enthusiastic lab. And I want to add that none of this can be accomplished without collaboration, including wonderful collaborations here at the University of Alberta. Andrei Kovalenko is in the audience, and Nikolai Blinov is in the audience. But none of this work can occur without grant funding. And an important source of our grant funding is through the Canadian Institutes of Health Research, but also through the Alberta Prion Research Institute. And I just want to underline that APRI will be going up for renewal in 2015. This is a stellar organization which has brought Alberta into the very forefront, the worldwide renown of of innovative approaches not only to prion disease, but to propagated protein misfolding in all of these neurodegenerative diseases I was talking about. I hope you will speak to your MLAs or whatever you can do to make sure that this outstanding organization continues to supply funding. So I thank you for your attention. I think that'll be it.